G-protein-gated inwardly rectifying potassium channels, or GERK, are important for regulating the excitability of neurons. GERKs can be activated by both G-protein-coupled receptors, GPCRs, such as the muscarinic receptor M2R, and by alcohol. To study the effects of mutations in the GERK alcohol binding site on its activation, a single-point mutation is introduced into the GERK cDNA by site-directed mutagenesis. Purified GERK constructs are transfected along with YFP constructs into mammalian cells, and the transfection efficiency is assessed by fluorescence imaging. Whole-cell patch clamp analysis is then performed to compare the alcohol and G-protein-dependent activation.
This visual experiment demonstration will provide you with key information on how to study the function of ion channels. Today we'll be looking at a question of how alcohol activates GERK channels.
The site-directed mutagenesis technique shown here can provide insight into ion channel function. This technique can also be applied to other systems where a mutation in a gene was known to cause human disease.
Visual demonstration of whole-cell patch clamping technique is critical because there are many steps that require precision, proper electronic compensation, and practice for generating usable data.
The DNA used for transfection in this protocol is prepared as follows. First, GERK subunit DNA from PCR is directly cloned into the mammalian expression plasmid pcDNA 3.1.